So, today's lecture is the cell division, how cell divide. Lecture number 3. Basically, cell is divided either through the mitosis or either through the meiosis. Basically, there are two types of the cell. One is the somatic cell and other is the germ cell. So, meiosis is always take place in the germ cells, not in the somatic cells. While mitosis take place both in somatic cell and also in the germ cells. So why cell division? What is basically cell division? Why do cells do it? How cell division take place in cells? And why the cell division is important to me? These three are the most common question for everyone. So first of all, what is cell division? Basically, cell division is known as the mitosis or the meiosis. These are the take place in the regular body cells and keep cell living and growing. So the cell division is most important for the cell living and growing. Why do cell divides? Basically, cell divides. The larger a cell becomes, the more demands the cell places on its DNA. Cell When a cell is grow larger in size, basically it occupies the large space on the DNA. So it also has the more trouble moving enough food and waste across its cell membrane. So when cell size grow in size, so it is difficult to reach the food to the every part of the cell where it is required. Every living organism is the made up of the different kinds of the cells. In order to keep living, in order to keep living a organ or a human or an animal, it is compulsory that your cells must stay alive. If your cell is dead, you are also dead. So, in order for cells to keep living, they must divide and multiply. Why is cell division important? So first of all is the all living things are made up of cells. So if the cell can't divide, then we can't grow. How we grow? Due to the division of cells. When cells divide, the more the number of cells, the more the size increase. The second one is the cell. Why cell? Because the cell is the basic unit of structure and functions. Because without cell we are nothing. All cells are come from the pre-existing cells. Because a cell when grow in huge size, the cell divide itself into the two equal parts, which is known as the mitosis. So for that the cell division is most important. This is the phases of mitosis. Basically this is the interphase, the most important and the first one phase. Basically mitosis is started from the prophase. This is usually not included in as such mitosis, but this is the part of cell division. So we say that the mitosis start from here: prophase, metaphase, anaphase, and telophase. So what is the mitosis? So mitosis the basic phases of cell life is this is the parent cell, the prophase, the metaphase, the anaphase and the telophase. Then here is the cytokinesis take place in mitosis. So basically what is mitosis? Mitosis is the type of cell division in which a cell divides into the two daughter cell and the chromosomes numbers cannot change from the parent cells. So if the chromosomes are 44 in the parent cell, then if the cell divide here its offspring also contain the 44 chromosomes, not less than 44 or not greater than 44. So this is the so we said that these daughter cells are the exact copy of each other because these are the exact copy. DNA is not changed in this daughter cell because these are the autosome cells. Autosome cells are the body part cell. If our when we said that if we wound it from a knife, then this wound will heal in further time. That is only due to the mitosis action. Mitosis take place on that part. Cell divides, multiply each other, and cover the wounded portion of our body. So those cells have the chromosome number same like the parent chromosomes number. So you said that there is the starting of this mitosis. We may say that the structure of chromosomes and the interface. Basically, chromosomes are the. This is the chromosomes. This red and the blue are the chromosomes. So what is this chromosome? Basically, this chromosomes is a thread-like structure which is present in the nucleus of the. This is nucleus. This is a thread-like structure which are present inside of the nucleus of the cell. So chromosomes are the on the chromosomes there are the genes are the present on the this is a chromosomes and on this chromosome different genes are present. These genes code our different structures, like some genes are for the hair growth, some genes are for the hand, some genes are for the color, some genes are for the height, and some genes are for the different proteins. So far, while the DNA is made up of the sugar, a pentose sugar, a phosphate group, and the nitrogenous bases. So each chromosomes has two sister chromatid. This is the one and this is the second. This is the one chromosome with two chromatid. One chromatid and second chromatid. These chromatid are attached with each other with the help of this chiasmata. This point is known as the chiasmata. So the first one is the interphase. What is interphase? Interphase is the longest stage of the cell's life. The time is spent between the two phases, like the mitosis and again the second mitosis. We spend the cell time spent in interphase, in which it produces all the material which are required for the growth and prepare itself for the division. This is the part of interphase where the cell begins. Interphase is known as the G1 phase of the cell cycle, while this is the growth phase. So this is the G1 phase. In this G1 phase, the resting period, this is basically the resting period after first mitosis. So we said that a G1 phase is also known as the resting phase after the one mitosis completion. But we also said that in this the interphase or G1 phase, cell usually grow its size, a huge size. Then cell goes to second phase of the interphase, which is known as the S phase or the synthesis phase. In this S phase here, the DNA is duplicates, DNA is replicate, DNA replication start, DNA replicate itself more and more. Like here you have seen the one chromosomes with one chromatid, but here it is duplicated. Here it is duplicated, like the DNA are more in amount. So the next one or the third phase of the interphase is G2 phase. This is the checkpoint to make sure the DNA is correct or not. In basically G2 phase, cell prepare itself for the replication. From here then the mitosis start. So you have seen that only the little portion of the cell cycle is a mitosis, but while the other cycle is so much long and time consuming. So when we talk about the time, cell remain in G1 phase for 10 hours. Cell remain in this portion for 10 hours, while in S phase, cell remain in S phase almost 9 hours, while G2 phase is about the 4 hours. G1 phase is 10 hours, S phase is 9 hours, and G2 phase is 4 hours. The first one of the cell mitosis is the prophase. In prophase, cell begins the division process. The nucleolus disappears. This side, the nucleus outer portion, nucleus is disappearing and nuclear membrane break apart. Like you have observed, this is the inner parts or the portions. Basically, this is the sign of disappearing of the nucleolus. While in case this is early prophase in which the nucleolus start disappearing, nuclear membrane breaks down, and this is the late prophase in which the chromosomes are visible. The spindle apparatus form is attached to the centromere of the chromosomes. Chromosomes are visible in prophase. Then the next one is the metaphase. In this phase, a tube-like structure, nuclear membrane is completely gone. You have easily observed the chromosomes, duplicated chromosomes line of the cell equator, and chromosomes are arranged in equator plate. Equator plate is the center of the cell. In the center, the chromosomes arranged itself on the metaphase. As you observe this one, and the spindle apparatus is observed. These two centrioles are formed on the left and right part, top and bottom. These are send the spindle fibers which are attached to the chromosomes. Chromosomes are attached with the spindle fiber and arrange itself on the metaphase plate. While in case of anaphase, this is the third phase of mitosis. The diploid sets of the daughter chromosomes separate with each other. They are pushed and pulled toward the opposite poles of the cell. Like the chromosomes are pulling from the centriole positions. This centriole pull the chromosome towards itself, which separate the two chromatids from each other. Like this one. Here you clearly observe the half chromatids are moving toward top pole, half chromatid moving towards bottom pole. While the telophase is the last phase of mitosis, in which the nuclear membrane and nucleolus reform, nuclear membrane starting reform, and the chromatids reach to a pole, this one and this one, chromatids reach opposite pole, nuclear membrane start forming then the cytokinesis take place. Cell plate begins to form, cell prepared for the final division. So the final stage of cytokinesis is in this cytoplasm organelles, nuclear material are evenly separated and two nuclear cells are formed. In cytokinesis, basically, when the in cytokinesis, the cytoplasm and other cell organelles like mitochondria, vacuole, Golgi bodies, these are formed in the cell, and cell divide into the two equal parts. These are the two new daughter cells which are exactly like each other. So we're moving toward the quick review. So first of all, in interphase, cell spend the majority of its time growing and function. During its S phase, the cell cycle replicates its DNA in the anticipation of the mitosis, which is the requirement of basically mitosis. While in early prophase, these are the two centrioles are emerged, and the chromosomes, which are two consisting of the two chromatids, are appear in the prophase. While in late prophase, these chromosomes are visible, nucleolus disappear, spindle fiber are attached with the chromatid. While in metaphase, these chromosomes arrange itself on the equator or the metaphase plate. In anaphase, these two daughter chromosomes are separated with each other, two chromatids separated with each other and moving towards the opposite pole. In telophase, these are reached to its opposite poles and cleavage furrows start growing, which is ultimate the separation of both cell. In cytokinesis, the other cell organs are developed. Cytoplasm is divided into the two equal parts. So anyone said that tell us the order of this diagrams. Which diagram comes first? Which is basically the interface. Which is basically the interface. No one?
C, sir.
Yeah, right. And then
E1.
right. After E, after E,
Mm, sir, A,
A, right. Then
then D, or then B.
A के बाद क्या बताया? सॉरी,
Sir, D,
सही है. Then
with
B.
B. Yes, sir.
सही है. Thank you. So this is the right order. This is the interphase, prophase, metaphase, anaphase, and telophase. So what is interphase? In this interphase, basically three phases: G phase, S phase, and G2 phase. In G phase, cell grows its size. In S phase, cell synthesizes DNA. In G2 phase, cell prepares itself for division. While in case of prophase, nucleolus and nuclear membrane start disappearing and the chromosome becomes visible. In metaphase, chromosomes arrange itself on the metaphase plate with the help of these spindle fibers, which attach to the chromatids. While in case of anaphase, these are moving towards the opposite poles. In telophase, these chromatids reach itself on the opposite plates, opposite poles. I think this video helps you.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
So this is all about the mitosis or the today's lecture. Video already uploaded on YouTube. No need to worry. So attendance. किसी का कोई क्वेश्चन है तो बता दें.